This is Dr. SRIDHARAN from SNS College of Technology. In this video, we are going to see about the UV visible spectroscopy for the analysis of organic and inorganic chemical samples. Introductions about UV visible spectroscopy. Ultraviolet visible spectrometer is another analytical instrument that measures the amount of UV and visible light that is absorbed by the sample, either organic or inorganic samples. It is on a widely used technique in chemistry, biochemistry, and other fields, especially for the pharmaceutical chemistries, to identify and quantify compounds in a variety of samples. It works by passing a beam of light through the sample and measuring the amount of light that is observed at each wavelength. The amount of light observed is proportional to the concentration of the observing component in the sample. The working principle of UV spectroscopy. Basically, spectroscopy is related to the interactions of light with the matter. Here, we use it for the different sample from the organic and inorganic samples. As light is absorbed by the sample, like organic or inorganic substances, the result is an the increase in the energy content of the atoms or molecules. When ultraviolet radiations are observed, this results in the excitations of the electrons from the ground state towards a higher energy state. The molecules containing pi electrons or non-bonding electrons, that means n electrons, can absorb energy in the form of UV light to excite these electrons to higher anti-bonding molecular orbitals. The more easily excited the electrons, the longer the wavelength of light they can absorb. There are four possible types of transition. Electron transition, that's a pi star, n pi star, sigma star, and n sigma star. So, and they can be ordered as follows: based on the intensity, sigma star greater than n sigma star greater than pi star greater than n pi star. The absorbance of ultraviolet light by a chemical compound will produce a distinct spectrum that aids in the identification of the sample. This is the modern instrument of UV visible spectroscopy, and also represent the schematic diagram of the UV visible spectroscopy, the light source, and passing through the lens, and then a sample, then the detector. So these are the steps we should follow for analysis of inorganic and organic chemicals. Parts of UV spectroscopy: light source, monochromator. Here the tungsten filament lamps and hydrogen deuterium lamps are the most widely used suitable light source, as they cover the whole UV region. The tungsten filament lamps are rich in red radiations. More specifically, they emit the radiations of 375 nanometer, while intensity of hydrogen deuterium lamps follows below 375 nanometer. The monochromators generally are composed of prism and slits. The most of the spectrophotometers are double beam spectrophotometers. The radiations emitted from the primary source is dispersed with the help of the rotating prisms. The various wavelengths of the light source, which are separated by the prism, are then selected by the slits. Such a rotation of the prism resulting in a series of continuously increasing wavelength to pass through the slits for recording purposes. So here the modern equipments automatically it moves to the different um, intensity, and that selects based on the our samples. And sample and reference cells, one of the two divided beams is passed through the sample solution and second beam is passed through the reference solutions. So double beam technique is the results are very accurate and we can get it for the better resolution. The both sample and reference solution is contained in the cells. These cells are made up of either silica or quartz. That is a transparent glass vials. Glass cannot be used for the cells as it also absorbs the light in the UV radiations. We are used for the special glass. That means it will not absorb the light. Detector, generally two photocells serve the purpose of the detector in UV spectroscopy. One of the photocells receives the beam from the sample cell and the second detector receives the beam from the reference. So both results are interpreted and we can get for the results. Then amplifier for the tune up and get it for the clear picture about our sample responses regarding the devices. We can get it the different responses based on our sample and the function groups. Applications of UV visible spectroscopy. It can be used in the quality control for production industry, chemical production industries, and pharma industry, cosmetic industry, petrochemical industry, pharmaceutical research, optical components, food and agriculture, life sciences, and the basic chemistry research in various areas, microbiology, material science, analytical chemistry, chemical and biotech industries, etc. Thank you.